Thanks, Emily, and thanks everyone for joining today. Good morning, good afternoon. Apologies for my slightly croaky voice at the moment. Hopefully I can get through to the end. So I'm going to talk to you today about off-target safety, and this is really about small molecules. So just a bit about me. I'm Vice President of Quantitative Systems, Toxicology and Safety at Certara, or QSTS. And my background in the initial part of my training was in academic research and in physiology and pharmacology. So that's what I was trained as a pharmacologist with a good grounding in physiology. So prior to joining Certara, I was in the pharmaceutical industry and specializing in safety pharmacology. So I worked for Syntex, Quintiles and AstraZeneca, joining Certara 5 years ago to head up the newly formed QSTS team, who are shown in the picture here. And our focus is on developing in silico tools to enable evidence-based decision-making in non-clinical safety. So I like this quote from W. Edwards Deming, who was a data scientist in the US. Without data, you're just another person with an opinion. So we want to move from an opinion-based to a data-based approach. So off-target safety. So here this diagram shows that a compound or its metabolite has a primary target, shown here in green, and both it and often its metabolites can act on secondary targets, and they can be of a similar type of receptor. And we're using a receptor here in the general sense of the term to mean a macromolecule that a drug binds to elicit its effects. So it can bind to a similar type receptor, so GPCR, or also, as shown here, or for example, other types of receptors, ion channels, transporters, enzymes, kinases, and nuclear hormone receptors. And the point to make is that in terms of the primary target, there's very little you can do about primary target-driven safety issues. You either have to accept them and manage them, or reject the target completely as being unsafe to go for. However, if you address secondary target-driven or off-target safety issues before selecting a candidate drug for clinical development, there's quite a lot you can do. You can basically design it out of the chemistry. So most pharmaceutical companies screen their compounds of interest against a broad panel of off-target receptors. We don't think that best use is made of the data, and the readouts can potentially be used both prospectively to predict outcomes in non-clinical and clinical studies, and retrospectively to explain findings in non-clinical and clinical studies. And our aim was to improve interpretation using an evidence-based approach. So this is a typical readout that you'd get from a screening company. And what they've done here is to arrange the receptors in descending order of affinity. So here the affinity parameter is either the Ki from radioligand binding or the IC50, and you can see a table here. They won't know this, but in this table you've put your predicted, because it is predicted at this stage, free Cmax plasma concentration that you're expecting to see in clinical use. And what the person responsible for secondary pharmacology has to do is to eyeball these and see which might be a problem if you go into the clinic with this compound. So what you can do is to plot this out. So this horizontal axis here is concentration on a log scale. The vertical line is the predicted free Cmax, and then these are the Ki or IC50 values from this table plotted out on the graph. So you might think, okay, well, some of these close to this free Cmax concentration might get hit by the drug in clinical use. So our question was, what if you could do this, which effectively color codes the hits according to the likelihood of a meaningful interaction in clinical use. And you can see here that it's not just a simple case that the ones that are nearest the predicted free Cmax are red, and the ones in the middle are orange, and then or amber, and then green. It depends on the receptor. So how could you do this? How could you go about it? So before explaining how we went about it, just knowing what potential side effects are associated with each of those off-target hits is not enough. And what you really want to know is, how hard do I have to hit this receptor to see these side effects? Or look at it another way, how much engagement with this target can I get away with before I start to see these side effects? And so this is about occupancy, and this is going to vary between receptors and between pharmacodynamic effects of a drug at the same receptor, perhaps in different tissues. And it requires knowledge of how much target engagement is required by the marketed drugs targeting this receptor by evaluating the ratio of their effective plasma concentration, unbound, divided by the Ki or IC50 at the receptor. And then you can compare the same ratio for your compound and ask the question, does it sit within the range of marketed drugs, or do you have a margin? So there is prior art in doing this, and this slide summarizes some publications that cover, there are 6 publications out there over the last 20 years covering 5 receptors. So I'm going to briefly go through each of these in turn from the top downwards. So the first one was from a group at Pfizer, and they looked at the ratio of hERG IC50 over the unbound plasma Cmax for marketed drugs to look for whether that would predict torsade de pointe risk. So generally speaking, drugs that were torsadogenic have got small ratios of this of hERG IC50 over Cu, generally below 30-fold, whereas mainly non-torsadogenic drugs have larger ratios, but you'll see a couple of torsadogenic drugs in there with larger ratios. But here's a principle where you can get some decent prediction just from hERG IC50 and knowledge of the plasma concentration. So I was involved in a larger study with about twice as many drugs, because this was about a year after their publication came out and there were more drugs available in the literature. So generally speaking, in this red box, these are torsadogenic drugs, and this is again the same scale, a log margin. So this dotted line here is a 30-fold margin, and so the majority of torsade producing drugs had a margin less than 30-fold, and in the green box the majority of safe drugs had a margin over 30-fold, and you've got some in this group that have got a very low incidence, which is you've got margins all over the place. But it's the beginnings of getting some predictivity of torsadogenic risk if you push the margin out. And in our paper, we advise going above 100-fold in future drug projects. Moving on to a different off-target receptor, this is the NaV1.5 or cardiac sodium channel. And if you block this channel, you prolong QRS duration in the ECG. So these drugs in the red box prolong QRS, and they've all got relatively small margins. So this is this band here is 30 to 100-fold, and then these are non-QRS prolonging drugs, and the vast majority have got larger margins. There's the same principle as was used with hERG and torsade de pointe. Moving on to a different receptor altogether. ENT1 is an adenosine transporter. So this group of authors were looking at the risk of dyspnea. So drugs associated with a high risk of high incidence of dyspnea have got a small ratio of ENT1 IC50 over unbound plasma concentration, whereas drugs not associated with dyspnea have a large. And again, here's the scale on a log scale here of the ratio. And there are some, rather like our study with torsades, there are some with a low incidence that are kind of somewhere in between. This is a smaller study, but it's from a group at the FDA, and they were looking at risk of valvulopathy or valvular heart disease with looking at the ratio of 5-HT2B Ki over plasma concentration unbound. So again, the ones in green are drugs not associated with valvular heart disease, and they've got large ratios, whereas the ones in red have got smaller ratios, the same principle applying. And finally, this group flipped the ratio, so they're looking at the opposite of Cu over IC50, and they were looking at off-target hits at VEGF2 receptor and elevation of blood pressure or hypertension. As you can see, drugs associated with elevation have a higher ratio. So here, because the ratio's flipped, this is about occupancy of the receptor. So there's 6 examples, 5 receptors. They've used the same principle as the ratio of Ki or IC50 to Cu, or the inverse of this ratio, Cu being unbound plasma concentration, as a surrogate indicator of in vivo receptor occupancy required to achieve an undesirable pharmacodynamic effect. However, they've used a variety of approaches, and the data curation criteria and data presentation differ as well. So what we've done is start again and apply a consistent approach, and we've taken it from 5 receptors up to 100 receptors so far and put it into a user-friendly software package. For each receptor, we expertly curated lists of reference drugs, which I'll walk you through in a minute. And these are defined as drugs that target that receptor for their therapeutic efficacy, so they're designed to hit that receptor. Most are marketed drugs, but also this includes candidate drugs either still in clinical development or discontinued, but basically they've got clinical data. So what we were looking for and then expertly checking was data on Ki or IC50, receptor selectivity, lowest effective plasma concentrations, plasma protein binding data, and side effects in clinical use. So we would start with a long list here, if you like. So you might end up with, I've just gone through letters of the alphabet, so 26 drugs here with Ki or IC50 data, and these have this receptor as their primary target. And then as you go through the process, you get, if you like, attrition from your original long list. So then you look for which of these have got clinical data available. A lot of them won't have made it into the clinic. And so what we're looking for is pharmacodynamic and adverse effects and plasma concentrations. And then finally, how many have plasma protein binding data available, because without that you can't calculate unbound or free concentration. So you might end up with maybe 6 or fewer drugs. And then expertly review and QC check all the data. So for each reference drug, so at receptor X, you've got one of the reference drugs. You know the molecular weight, which you can get from Kemble, for example. Got plasma protein binding data. You've got the Ki at the receptor in micromolar, and you've got the effective plasma concentration which would be originally in probably nanograms per mil, and you're converting it into micromolar, and also taking into account the plasma protein binding or the free fraction to calculate unbound concentrations. That's what you're working with, are the same units. So we're looking at the ratio of C effective plasma concentration unbound, which we also abbreviate to Cu, over Ki or IC50, depending on the assay receptor type. And then you plot this out. So here's the scale of the ratio of Cu over Ki, and this is drug A from that list. And then you do the same for all the other drugs in the list, and you'll get a spread something like this. But the drug of most interest to us, or the pivotal one, is this one here, which is the one with the smallest or the lowest ratio of Cu over Ki. And that determines where the high risk category, these are relative risks, but this is what we class as or color code as red. We've got an arbitrary 1 log width band to the left of that, and then beyond that is green. So that's what it looks like in a PowerPoint slide. This is what it looks like in the software, which is a bit more compressed here. So here are the reference drugs. This vertical line is the ratio of 1, where the Cu is equal to the Ki. And if you like, this is reflecting increasing receptor occupancy as you go from left to right. This point here is a test compound which has been plotted in the software, and its position indicates that it's deemed to have a low likelihood of causing side effects via this off-target receptor in clinical use, as the receptor occupancy is below the level required to elicit an effect. So there are 3 outcomes, and this is the same compound tested at 3 different receptors. So at receptor X, it's come out as green, and we would recommend further investigation being unnecessary. If I jump to the red outcome in this receptor, a red outcome doesn't mean stop the compound, it means pay attention. So you would consider further investigation of the potential safety issue, which could involve in vitro and or in vivo studies, which may involve additional endpoints in a safety pharmacology or toxicology study, or you might have to do some bespoke specialized study to capture the effect. And the one in the middle, again, this is a negative outcome. It's outside the red zone, so it shouldn't impact on this receptor. However, what you've got to watch out for is if your estimated or predicted free Cmax increases, which often happens during drug development, then that could encroach into the red zone, and I'll show you a slide later to show how you can look at what if scenarios around that. So this is our list of 100 receptors. The receptors in blue are from the Bose panel, which is a consensus panel from 4 pharmaceutical companies published in 2012, comprising 44 receptors. So we've got an additional 56 receptors in our panel. Now, you know, very few companies screen at 100 receptors, but the idea is that they tend to have the Bose panel or most of it in their screen, and then if they're using our software, they can pick from the remaining receptors to build or match to their panel. We're also working on these 8, I think it's 8 or 7 additional receptors here. And further receptors will be prioritized by feedback from users. So in numbers, you've seen 100 receptors, and you've also seen our starting point is over 3,500 reference drugs, from which over 400 are plotted. They have the full data set required. I'll come on to mechanistic safety pathways, but we've got over 200 of those. And it was the work of over 6 FTEs for about 3 years. Not shown here, but the software will auto-generate a word report for your compound, and it can be accessed either by purchasing a software license or by consultancy with us. And this is just a word cloud of the commonest side effects, perhaps no particular surprises in there. So I'm just going to show you some screenshots from the software. So this is our receptor panel view, and the receptors here are arranged by class. So these are the GPCRs, ion channels, nuclear hormone receptors, enzymes, kinases, and transporters. If you click on one of those receptors, it takes you to an individual receptor interaction page, as shown here. So what we've got in this box here is the safety implications of interacting with this receptor in humans and animals. And you'll see that the table is divided into two columns here. So this is a positive modulation, so for GPCR, an agonist effect, for example, and then this is a negative modulation. So for GPCR, it would be antagonism or inverse agonism. For an enzyme inhibition, transporter inhibition, ion channel block, etc. And there is a link against the name of the receptor to our UFAR, the IUFAR BPS page on that receptor that will tell you more about it. And then in this panel here, there's a list of drugs targeting the receptor, and the ones in bold are plotted on the right, which we'll come to in a minute. And there's links to IUFAR here and Kemble, so you can find out more about each of these drugs and compounds. Oh yeah, we also list the endogenous ligand at this receptor. And yeah, on the right hand side, a list of tool compounds. So these haven't been in human beings. There are some veterinary drugs as well in here. And this is useful if you wanted to set up an assay to evaluate the functional effects of this interaction. And you've got some comparator compounds you can choose from, some of which are less expensive to buy than others. So on the right hand side is the plot that you saw earlier, and as I stated earlier, the position of the start of the red zone is set by the reference drug with the lowest Cu over Ki ratio. So this is the most stringent setting, if you like, and that's quite common in tox assays, that you'd go for the, if you like, the worst case scenario. And that, just repeating what you saw earlier, this test compound is deemed not likely to interact with this off-target receptor in clinical use at the expected plasma concentration. Now that's quite a useful outcome, because often if you get fixated, or one of your colleagues gets fixated on one of the so-called hits, hours and hours of discussion are wasted on it, and secondary intelligence helps you to triage the outcomes and deprioritize these green outcomes, so you don't waste endless time debating it. So there's also, as I mentioned earlier, a what if scenario option, and this is where you can change the expected or predicted unbound plasma concentration by using a scale factor. So if you change it from 1 to say 10 times the actual CU you've entered, you get a second plot. So here the test compound's been moved by that amount to the right. So it still won't, even if you, in this case, if your predicted free Cmax is underestimating what you end up with by tenfold, you're still not hitting that receptor. However, if you had been in the amber zone to start with, it would then push it into the red zone. So it's quite a useful function to have in the tool. And then there's a link at the top to mechanistic safety pathways. So if you click on a mechanistic safety pathway, and this refers to the receptor page we've just seen, they follow a format that should be familiar to you if you have seen any adverse outcome pathways in that are used particularly by chemical toxicology groups, as opposed to pharmaceutical toxicologists. We didn't want to call them AOPs because there's a lot of sort of baggage that comes with that, but we've used the same format. So we've got, it starts on the left with a receptor interaction, and then you describe what happens at the cellular level, at the organ level, and then the whole organism. The value or the advantage of using this kind of diagram is that you're obliged to use the same diagram each time across the receptors and across the mechanisms. Whereas if you had a document explaining this, some of them will be about 3 lines because you don't know much about it, and others would run to several pages. So it's keeping them constrained within the same format, and it's backed up by references, and we've gone for the minimum number to get you started. So this is one example, and it's alpha-1 adrenoceptor, alpha-1A adrenoceptor antagonism causing postural hypotension. I'll go through it because I'm going to return to this with some proof of principle data. So at the receptor level, antagonism of post-junctional alpha-1A adrenoceptors on arterial smooth muscle cells leads to, at a cellular level, pharmacological antagonism of smooth muscle contractile response to sympathetic vasoconstrictor mediators, noradrenaline or norepinephrine. And then at the organ level, there's attenuation of the postural reflex vasoconstriction. And then on standing, blood pressure decreases, which is known as orthostatic or postural hypotension. Another example, and again I'll return to this one, is HERG block leading to QT interval prolongation and torsade de pointe. So again, block of HERG on cardiac ventricular myocytes leads to reduced outward current and prolongs the action potential duration, leading to conditions where there's an increased risk of early after depolarizations, which are arrhythmogenic. The organ level, the QT interval is prolonged on the ECG, and there's a risk of seeing arrhythmias, and if sustained, they can lead to ventricular fibrillation and a drop in cardiac output as a result, with loss of consciousness and death. So those are the 2 examples from mechanistic safety pathways, which I'd like you to try and recall in a minute. The other point to make is that obviously it allows you to predict clinical side effects even before running any animal studies, and I'll go through the 2 examples I've just shown you in mechanistic safety pathways in a minute. Another point to make is that some side effects in humans are not readily or reliably detectable in animals, or they may require quite specialized techniques and large numbers of animals. And these are predictable with reasonable limitations and caveats using secondary intelligence, so including ones that are not readily detectable in animals, like abnormal dreams, nightmares, akathisia, dizziness, dysphoria, dyspnea, hallucinations, headache, and suicidal ideation. Side effects requiring specialized techniques. So these are detectable, but you have to set up quite specialized studies. I'll mention abuse liability in a minute. Anxiety, erectile dysfunction, fatigue, insomnia, memory impairment, nausea, visual dysfunction. So in my previous career, I probably did do studies on about all but 1 or 2 of these, and they're quite specialized techniques that you don't detect in the normal safety pharmacology GLP package. So on the abuse liability one, in the FDA guidance the document does mention prediction from receptor profiling, and they mention some receptor families here. So what we did was we looked carefully at the receptor families and what was on offer in abuse liability testing panels, and arrived at our list of receptors here across the different types of receptors. So we've got 35 receptors in there. And we've also included ones that are not in FDA guidance because theirs is focused on CNS psychoactive drugs. We've also included sports performance enhancing receptor targets that could lead to abuse. And another point to make for the CNS receptors is that secondary intelligence is based on plasma concentration because that's a common currency across all drugs. Very few have got free brain concentration data available. So it's really the worst case scenario because the free brain concentration will always be lower than this. So if you do get if you get a green hit, it's never going to trouble you in terms of effects on the brain. Whereas red hits, you can then further assess them. You can go in and say, okay, well, does my drug get into the brain? And so you look at the brain penetration or the predicted, if you model the brain penetration and factor that in. So it's an early flag. And remember that the in vivo studies for abuse liability, if you need to do them, won't be done until you know the dose level you're going to be using in clinical use. And so that's kind of late phase 2. That's quite a long time to wait. So this is quite a good early warning system. So I'm going to go through these two studies, and I'll call this proof of principle. I wouldn't call it validation really, but it's a very difficult to do for two reasons. First of all, adverse effects are not uniquely related to a single receptor interaction. So you can go for ones that are at least restricted to a few receptors. So for example, QTC interval prolongation is closely linked to Hurg block, but there are other causes of it. And likewise, postural hypotension can have other causes. The second, there's probably three reasons. The second reason it's difficult to do is you need a large number of drugs with off-target Ki or IC50 data, quality data at the receptor of interest. And thirdly, the other challenge you've got is those drugs that you do use, you don't know what their other pharmacology is, apart from maybe their primary target, obviously, but across the spectrum of targets, you don't know what else they're doing. So you've got to bear all that in mind. But we had to go at two receptors. So the alpha-1 adrenoceptor, as you heard earlier, is located on arterioles, and noradrenaline, norepinephrine is released continuously from sympathetic nerve endings and docks onto the receptor, maintaining cell contraction. Meanwhile, blood pressure in the carotid arteries, in particular carotid artery, carotid body baroreceptors, carotid sinus baroreceptors, is monitoring the blood pressure for the blood supply to the brain. So it's particularly interested in drops in blood pressure. So if they detect a decrease in blood pressure, there's a reflex increase in the firing rate of the sympathetic nerves, releasing noradrenaline onto blood vessels. So this typically occurs when you stand up and you get a vasoconstriction. So in this diagram here, the subject is being lying down, stands up, there's reflex vasoconstriction, blood pressure is maintained, the subject is completely unaware of anything going on, everything's normal, stand up, no ill effects. If you block the alpha-1 adrenoceptors, and you see a little red antagonist in here blocking the receptor. Then the subject stands up. That vasoconstriction as a reflex response is impaired. So there's still a reflex increase in firing rate of the sympathetic nerves, but the noradrenaline effects are being impeded by the antagonist, and the subject feels dizzy. Feels dizzy because there's a drop in blood pressure due to gravity. So this is not a trivial side effect, particularly in elderly patients, because they can, when they faint, they may injure themselves. That could fracture their pelvis, for example, or other fractures which could result in hospitalization, or even they could fall down the stairs. So, you know, this is not a trivial side effect. You don't want it as a side effect with your drug. So we had access to a publication which had tested quite a lot of antidepressants and antipsychotics and other drugs we got from literature, and had run Ki values at alpha-1A adrenoceptor. So it's not their primary target, it's an off-target. So this table shows drugs with a strong association with postural hypotension in clinical use, and this was the outcome in secondary intelligence. So we set in secondary intelligence, you can set it to the 3 color outcomes of red, amber, and green, or you can remove the amber outcome, which is what we did for this binary outcome here, just had red or green. So the majority were correctly predicted by secondary intelligence. We did have 4 false negatives that really don't interact with this receptor, but they still cause postural hypotension. So this comes back to the fact that really we don't know what else these drugs are hitting. We could do with a profile really of them, but that doesn't exist in the literature. But they're most probably causing postural hypotension via a completely different mechanism from this. But I think a sensitivity of over 80% is pretty good going for what is effectively an in vitro, assay Ki value. And then these drugs, a similar number, same number, 23, had a weak association or no association with postural hypotension in clinical use. And again, secondary intelligence correctly predicted most of them, with some false positives. So there were 5 that had false positives. And remember that, first of all, the plot of the reference drugs and where the red zone starts is at the most left-hand drug, which is the most stringent. So we've set a tough criterion for test compounds. So it could be that it's because of that, or they may possess other pharmacological activity that offsets this effect on the alpha-1A adrenoceptors. But again, I think we do it with this tool, as with most in vitro tox assays or safety assays, they tend to be set with a high sensitivity and a lower specificity, and that's what we found here. So the specificity here is lower than 80%. If we move on to the next one, which is HERG. So as you know, ICH-E14 has required all small molecule candidate drugs to be assessed in a thorough QT study, or TQT, in healthy volunteers or healthy subjects in phase 3, with certain exemptions, for example, oncology products. So the TQT protocol considers an increase of QTC interval of greater or equal to 10 milliseconds, which is tiny, to be TQT positive and indicative of a torsadogenic risk, which appears on the drug label. So there was a revision in 2022, along with ICH-S7B, which was the non-clinical data, and this allows waivers in certain circumstances. to get a wave, you require double negative non-clinical data, so negative hERG and negative in vivo QTC. So this paper by Hugo Vargas from Amgen and colleagues across the industry proposed a hERG margin of greater or equal to 30-fold as being a hERG negative. But it's likely that if you're trying to predict a 10 millisecond increase, that you might need a larger margin than that. And this is what we set out to achieve. So in our hERG plot, as you can see here, these are plotted with the as Cu over IC50 at hERG, and the Cu is the unbound plasma concentration associated with a 10 millisecond increase in QTC interval. And particularly this drug here, which is more or less the left-hand drug, dofetilide, has got an abundance of good quality PK/PD data in humans, and also a large number of IC50 valuations which we can take the mean of. So we're quite confident about dofetilide setting this red zone. So a negative drug would come out here in the green zone and positive there. So this is what I meant by setting it as a binary evaluation. So we looked at, first of all, this is a list of drugs that it was constructed with that you've just seen. These drugs have all been through a TQT study, thorough QT study with publication. So they've got plasma concentration, and they were TQT positive. They prolonged the QTC interval by more than 10 milliseconds. And then this group were tested and were negative in the same protocol. So we looked first of all at the TQT positive ones and put them through secondary intelligence using the plasma concentration used in their TQT study. And as you can see, the majority were correctly predicted, 8 out of 9 drugs. The one that wasn't is perseriotide, which is a 1 kilodalton somatostatin analog, which causes bradycardia and hyperglycemia, which may contribute to QTC prolongation by non-hERG mechanism. So we thought this was pretty good on the positive side, so high sensitivity. On the negative side, 4 out of 7 correctly predicted, so the specificity is lower, as is often the case, as I mentioned earlier. So we'd be quite happy with that, because if you've got a hERG positive, like you've got with these 3, it's possible that they might block CAV 1.2, calcium channel in the heart, which offsets the effects of hERG block in prolonging QTC. We don't know that because there's no publications on it, but that would be the next step for your compound, and then do some modeling of effects on cardiac action potential. But I think for a tool just using hERG IC50 and the predicted free Cmax, this is a pretty good outcome. So that's just a summary of the hERG proof of principle. One point to make is that in the case of oncology drugs which don't require a TQT, a negative outcome in the hERG plot in secondary intelligence provides confidence that the candidate drug is safer than all the reference drugs, which are all QTC prolonging class 3 antiarrhythmics. So just to finish with potential values. So these first 2 rows are about interpretation and contextualization of secondary pharmacology readouts, either in the discovery phase or on the candidate drug, and it tells you which off-target interactions to focus on and which ones to basically leave to rest in that they're in the green zone. So that's predicting outcomes. Also, you can use it retrospectively, where you've seen an in vivo finding and you're trying to identify the culprit off-target interaction and rule out ones that are not likely to be contributing. And then finally, for large pharma, if you're in licensing compounds during the very brief and very important and intense due diligence activities, it would highlight potential issues which may or may not have been addressed in animal studies. So in summary, we have addressed 100 off-target receptors that are incorporated, including the Bose panel of 44 receptors. It's not possible to guess at what level of occupancy is required for an off-target effect for a given receptor. And the tool enables you to achieve consistency in secondary pharmacology interpretation across receptors, across compounds, and across projects, and also across users using an evidence-based approach. And so I'd just like to thank my colleagues in Certara and outside Certara for this work, and thank you again for dialing in and for your time, and happy to answer questions or try to.
Thank you, Will, for a wonderful presentation. If you have not already done so, please feel free to submit your questions in the control panel so that Will can answer them here live. It looks like one has already come through, so we'll start with that one. What if we only generate % inhibition data in our secondary pharmacology screening? Can we still use secondary intelligence?
Okay, so this is a good question, which I've got a couple of slides on actually in the spare slides. Some pharmaceutical companies will screen at a fixed concentration, usually 10 micromolar, and look at % inhibition. And the CRO doing the screening may use a sort of cutoff to decide whether it's a hit or not. It's usually 50% inhibition. And we know from radioligand binding assays, etc., that you can estimate the IC50 if you get 25% inhibition or more. You can probably push this down to 20%, but by this stage it gets a bit difficult to predict. But you're on the linear part of the sigmoidal curve with 25% inhibition or more. Between 25 and 75% inhibition, you can predict the IC50. So if I just show how to do this, you have to assume that the hill slope, so that's the slope of the effect concentration curve, is 1. And this is the Hill equation here that you rearrange to find the IC50. So X is the test concentration and Y is % inhibition. So it's quite simple to predict IC50. And then from the IC50, you can use this equation called the Cheng-Prusov equation, and that also requires the concentration of the hot ligand, the radiolabeled ligand, and its dissociation constant. So you can find that out and calculate the Ki. And once you've got the Ki, you can use secondary intelligence. So we can send you formulae in Excel to do this, and we're planning to put this as part of the data entry feature of secondary intelligence. But yeah, it's a good point, because some companies will go all the way to generating Ki values and others will stop at % inhibition.
So thanks, Will. The next question that came through is, can you expand on situations where the off-target receptor is located in the CNS, and how do you take into account brain penetration of our test compound?
Yeah, I kind of mentioned that earlier with respect to abuse liability, but it applies to all receptors that are present in the CNS. There's only a couple of parts of the brain that don't have a blood-brain barrier, area postrema being one of them. But so we're talking about the majority of the brain. Most drugs are kept out to a large extent. So the plots are built using plasma concentrations of reference drugs, those reference drugs targeting CNS receptors by their nature are pretty good at crossing the blood-brain barrier, whereas your test compound, unless it's specifically targeting a CNS disorder, generally speaking, has got a low brain penetration. So the free plasma concentration can't exceed the free brain concentration. So the free plasma concentration is kind of a worst case scenario. So if you get a red hit, you've effectively got to look at the brain penetration of your compound and compare it with, if you can find it, the left-hand drugs brain penetration, which will be obviously more than yours. And so effectively speaking, if you get a green outcome, you don't need to do anything more, or the same goes for an amber outcome. It's when you get a red hit, it's then just doing some simple back of the envelope calculations and figuring out whether you're still in the red if you take into account the brain penetration of your test compound compared to the reference compounds, reference drugs.